our next speaker is Sudha Yellapantula, PhD student in Benama Zhang's group. She's interested in understanding the organization of human language in the brain for the benefit of people with language disorders. Her work involves combining ideas from data-driven causality, information theory, and graph theory, and applying them to the analysis of brain data. And today she's going to be talking about information and graph theoretical machinery for neuroengineering. So,
thank you.
Welcome, Sudha.
Thank you. Hello. Yes. Thank you for that kind introduction. We humans are remarkable in our ability to produce sophisticated language with grammar and lexicon. Unfortunately, millions of people every year develop language disorders because of brain injury or stroke, where a part of their brain dies, causing lesions. And these lesions lead to many speech and language disorders, the most common one being aphasia. So here is an example. A person who develops a lesion in Broca's region, shown in green, can understand language, but they cannot talk. While a person who develops a lesion in Wernicke's area, shown in blue, they can speak fluently, but it is utter nonsense, and they cannot understand anything. Unfortunately, those are not the only two aphasias. We have many aphasias caused by lesions in a variety of sizes and shapes and locations. And aphasia research is hard. There's too much variability between patients, and there is no animal model that you can use to study language. So how would we develop new methodologies to help people who have aphasia and other language disorders? So, well, firstly, we need to understand how language is organized in the brain. What are the connectivity patterns of information flow in the brain? So, to do that, we have basically identified that we need to understand language before we can fix the system when it breaks. And we identified three main problems that we need to solve. So the first is we want to measure causality between brain regions during a task. Secondly, we want an analysis methodology that can give us spatial-temporal patterns of connectivity, again, within the task, on any language task. And finally, we want to relate neural responses that we observe with the connectivity patterns. So to do this, we need data. And we decided to go with ECOG data as it has excellent spatial-temporal resolution. Unfortunately, it is invasive. Well, luckily, we were able to collaborate with a neurosurgeon, Dr. Nitin Tandon, and he implants electrodes inside human brains as they're epileptic patients who are going for surgery. And while they are waiting for their seizure to be localized, they perform a variety of language tasks. And we are able to obtain data from those recordings. So here is the language task that they perform. This is a picture naming task. At time 0, which is stimulus onset, an image is shown on the screen, and the patient says, alligator. And that is one trial. And there are hundreds of such trials done. And from this data, we want to understand how language is organized in the brain. Before I show you that, I want to show you this, what currently exists. So the current state-of-the-art analysis is based on high gamma power, which is the power in the high frequency component of the ECOG data. And this has been shown to correlate very well with the task activities. So we did that in our own data, so I'm going to show you what that looks like. So this is a raster plot from one electrode in the brain, which is a visual cortex electrode. And I'm going to explain what this means. So the x-axis is time. We're starting from 0, stimulus onset. the image is shown. And the rows of this matrix are power in different trials stacked on top of each other. The trials are ordered in ascending order of articulation time. And you can see that there is an increase in power aligning exactly with when the image was shown for all trials. So this is basically a visual electrode. You see an image and boom, there's a power in the visual cortex. And the articulation happened a little later. So the trials are in ascending order. So that's why you see that shape of articulation time. Now you contrast that to an electrode in the motor cortex. This is where the power in the electrode occurred when the person started speaking. And you can see that it aligns very well with the articulation time. So in this way, this is 2 electrodes in the brain. And now I'm going to show you all the electrodes in 3 different time windows. This is the average power from the trials. And you can see that when the image was shown, there's an increase in power in the visual regions, which is at the back of the brain. And then when they're about to talk, there's an increase in power in the motor cortex, frontal, temporal regions. And after they talk, you can see an increase in power in the auditory cortex, which is 3 electrodes right there. And that is because the person heard themselves speaking. So you can see that you can track the task dynamics pretty well with high gamma power. Unfortunately, you do not know what the underlying connectivity is in this task. You do not know how information went from one brain region to another brain region, and that caused a power response. So we don't really know that. And so in our work, we try to address that limitation, and we developed a framework which we call DIMAS, Directed Information Multi-Scale Analysis, and we try to address all 3 problems that we discussed before. So firstly, we wanted to measure causality between brain regions, and we used directed information for that. We estimated directed information between a pair of electrodes using all the data from the trials in a small time window. And this is non-parametric, data-driven. So it doesn't assume any linearity. And by doing that, we were able to get an estimate of information flow in between one pair. And we repeated this for all pairs of electrodes and obtained a DI matrix. And then we normalized this DI matrix with a baseline. And now we say, if you see a red pixel in that matrix, that says that there was an increase in information between that pair of electrodes compared to baseline. So it's a task-related increase in information. And in this, here's an example of an electrode, which is a strong source. So you can see that it is sending information to a large number of electrodes. So yeah, in this manner, we were able to find causality between all pairs of electrodes. But now we want to understand the spatial-temporal dynamics. So we have a bunch of time windows, sliding time windows in the task, and we could obtain a bunch of matrices. It's very hard to just look at them and obtain dynamics information. So we went to the graph theoretic domain. We converted every graph to a brain graph. The nodes of the graph are electrodes of the brain, and the edge of the graph was the increase in DI compared to baseline. So in this manner, we now have a bunch of graphs, and we can do a multi-scale analysis on these graphs. Multi-scale, so we can have temporal dynamics information at different scales. So at a very broad scale, we looked at connection density. And we zoomed into an intermediate scale to look at clusters of information using the Louvain community detection algorithm. And then we zoomed in even finer to the node scale to look at the in degrees and out degrees, which would give you the best source and sinks of information. So I'm briefly going to show you the results of what we obtained. So this is a plot of connection density across time. So here, every graph was converted to one single point, connection density. And we found that there was a peak in connection density that aligned exactly with when a person was about to speak. And there was a peak in connection density aligned with when they heard themselves speak, as also aligning with the power increase in auditory cortex. So we believe that the first peak is when they found the word in the brain. So we think that connection density reveals temporal dynamics of the task. Then we looked at the Louvain community detection. in which we tried to pick out clusters or groups of nodes that were more connected with each other than with other nodes. And by doing this, we were able to find that in any given time window, we could find what were the best clusters that were in the brain in that, at that point in time. So here, right before articulation, we see that there was a Broca's region cluster, there was a motor cortex cluster, a frontal temporal cluster, and so on. We can also look at dynamics by looking at how these clusters changed with time. So here I'm showing you before articulation, we had 4 clusters. At articulation, we had 2 clusters. And after articulation, you can actually see a much stronger cluster over the auditory cortex. So at an intermediate scale, we obtain information through the Louvain. But when we zoom into the fine scale, we can tell you about every node how much of a source and sink it is. So in degrees is the sum of connections entering the node, and out degrees is the sum of the weights of the connections leaving the node. And the kind of information we obtain is, in a given time window, we can identify which regions were the strongest, here sinks, for example. In the right, when the image was shown, we found that temporal cortex were the top sinks of information. And when they were articulating, the motor cortex were the top sinks. But it is more than just saying that this region was the top sink. We now have information of which individual brain region, very specific brain region, that was the most active. So we have much finer resolution about the information dynamics. So in this way, we have uncovered we have a way to now obtain spatial temporal resolution of at different scales. And now you might ask, so how do these connectivity patterns and patterns of information flow relate with the power responses that I showed you before? And they got to be related because both are definitely related to the task. Both are giving us task information. So we looked for relationships between the fine scale analysis and the high gamma power, and we found a very significant correlation between the in degrees of a node and the high gamma power. We found both positively and negatively correlated electrodes. I'm going to show you what I mean by that. Here is a single electrode. The graph on the left, you see the red line is the high gamma power response over time of that node, and the blue curve is the in degree strength, which is the total information coming to the node. And you can see they were very highly correlated, 0.92. And similarly, we had a negatively correlated electrode where information came to the node and the power went down. So we found this not just in 2 electrodes, but actually a large number of electrodes in a single patient. And we repeated this for 7 patients, and we found this to be a phenomenon observed across all 7 patients. And this actually gives us an idea now that we see a high gamma power response, but we now know where the information was coming into that node, so we can identify which brain regions were driving that high gamma power to go up or to go down. So we have a much finer understanding of the language process at that patient-specific level. So in this manner, we are able to relate the neural responses we see with the underlying connectivity. So what are the next steps? So we think of an immediate next step that we think we can address with DMUS framework, and I'm going to walk you through that. So I'm going to try to use not too much medical terminology, but I'm going to explain what happens in the surgery room. So the neurosurgeons, their job is to resect the brain so the epilepsy, the seizures, they stop. And they don't want to resect a part of the brain that is doing something very important, like extremely language related. So they want to map language on the brain before they do surgery. And the way they do that is they have this grid of electrodes, and they ask the patient to do a bunch of tasks. This is just for mapping language, not for research. So they ask the person to read something on a screen, and they keep zapping electrodes with pretty high current. And when they zap a brain region, it causes a temporary lesion and a temporary aphasia. And when they zap a certain region, suddenly the person cannot talk. And they say, oh, that brain region is doing something related to talking. And then they ask them to read pictures, and they are no longer able to name an object, They said that is related to object naming because they couldn't name it. So this is the way in which in the surgery, before they go resect the brain, they figure out where the language is. This method has some problems in which it causes seizures and a discharge effect after you zap the brain. So we believe that these connectivity analysis from DEMAS, we already have the electrodes, the patient has already done those tasks for research, and we can develop these maps to localize language. So we could use these maps to help the cortical stimulation mapping they do, and maybe it's a safer alternative in the future. And the vision is that we can use these kind of connectivity analysis from DEMAS combined with cognitive theories to improve the language models that we have. And maybe in the future, we are going to be able to identify the correct brain regions to stimulate either invasively or non-invasively, and hopefully target people with aphasia who have no hope of recovery. So with that, I would like to acknowledge my advisor, Dr. Behna Majang, and our collaborator, Dr. Nitin Tandon. They're right across the street. It has been a wonderful collaboration with their team, and I am happy to answer any questions. Thank you for listening. Oh.
Thank you for a very interesting talk. Have you done the step where you actually compare your DEMAS to what they're finding in the surgical testing? Because I think that would be a really interesting correlation between the two.
That is the next step that we want to do, is to look at their cortical stimulation mapping. And when I did show all my results to the neurosurgeon, and he was very excited that, oh, these are the language areas, this is exactly what we expect. So he found it very intuitively satisfying what he thought.
I was just curious, did it happen to be that any of the patients that they were in there for epilepsy to get treatment for that, did they also have any of those aphasias that you talked about at the beginning of your surgery, and did you see anything interesting with that?
So actually none of the patients we analyzed for language had any language disorders, because we were trying to understand how does language work in normal humans. So we tried to pick the people who were most language normal. However, after the resection, there are some defects that show up. Some people do have some aphasias. Some people get better. Some people don't. So there's a percentage of side effects that do exist. Thank you. Thank you very much.